Now let's talk about pyruvate. Pyruvate is going to be an important intersection point for several metabolic reactions. We had a molecule of glucose, we created some pyruvates. Now, what are the pathways this pyruvate molecule can take? First, pyruvate can be converted to alanine in the liver via alanine transaminase, or ALT. You'll see ALT levels lots on clinic rotation, so now this is kind of the basis for it. This is how amino groups are transported from the muscle to the liver. Second, pyruvate can be converted to oxaloacetate via pyruvate carboxylase to either replenish the TCA cycle or for gluconeogenesis. Third, pyruvate can link glycolysis to the TCA cycle when it's converted to acetyl-CoA via pyruvate dehydrogenase. And then fourth, pyruvate itself can basically be the endpoint for energy production in anaerobic glycolysis, since it's converted to lactate by lactate dehydrogenase. Now this reaction regenerates our usable form of NAD+, so that glycolysis can continue to make ATP further. Anaerobic respiration is going to be the main source of ATP in red blood cells, leukocytes, the kidney medulla, lens of the eye, the testes, and the cornea. Do you remember where the Krebs cycle takes place within a cell? The TCA cycle, or the Krebs cycle, takes place in the mitochondria, and it produces high-energy electron carriers, NADH and FADH2, which then go on to generate ATP in the electron transport chain. Now this completes the metabolism of glucose. This diagram will show the basic steps of the TCA cycle. Notice how acetyl-CoA is fed into the system spins and creates various activated carriers. Now, we talked about the activated carriers earlier. It also produces a GTP molecule here, which is the equivalent of an ATP molecule because it can donate a high-energy phosphate bond to catalyze reactions, just like ATP does. So we think of it the same as an ATP. However, where have we seen GTP before? We got to think back to protein synthesis. GTP is used in the initiation phase of protein synthesis, where initiation factors help assemble the 40S subunit of the ribosome and transfer RNA. Now let's jump back to the TCA cycle. Once the cycle's done, the TCA cycle is ready for another input of acetyl-CoA, and all right, partner, keep on rolling, baby. You know what time it is. So we keep on rolling. Well, this is a very basic outline here of the TCA cycle. Now let's make it a little more complex by adding in some enzymes, throwing some intermediates to this cycle. Now you've seen this before in undergrad and in medical school, so it shouldn't be too scary to you, but let's cover some of the basics here. We start with an acetyl-CoA. Now how do we make that acetyl-CoA? We need to convert a pyruvate into our acetyl-CoA. Remember, we used our pyruvate dehydrogenase complex. So we created an acetyl-CoA. Now, as some byproducts, we created a CO2, and we also created an NADH in the process. So we can create pyruvate and convert it to acetyl-CoA, and that's one way we can start out our TCA cycle. Now, also, remember earlier I said everything is interconnected. We had that big chart that linked all the different reactions together. I said that carbohydrate, protein, and fat metabolism are all interconnected. Now notice how proteins and lipids can be converted into acetyl-CoA, which can then get fed into the system. So that's how we break down proteins and lipids. All right. So now let's start off with the net reaction of the Krebs cycle. The net reaction, we're going to produce 3 NADHs, 1 FADH2, a couple carbon dioxides, and a GTP, which is pretty much an ATP equivalent. Now this accounts for 12 ATP total. These activated carriers here will later exchange their electrons to make 3 ATP per NADH molecule and 2 ATP per FADH2 molecule. So since each glucose yields 2 acetyl-CoA, remember 1 glucose equals 2 pyruvate, and 2 pyruvate is equal to 2 acetyl-CoA. Each molecule of glucose will create 2 molecules of acetyl-CoA. Now this means that you get 24 ATP per molecule of glucose. The TCA cycle is going to be regulated at 3 major steps, which are going to be catalyzed by the enzymes citrate synthase, isocitrate dehydrogenase, and then the one that we've already kind of talked about, alpha-ketoglutarate dehydrogenase. Now, by the way, for the USMLE, you won't likely be tested on molecular structure, so that should take some stress off your chest. However, you should remember that the steps of the TCA cycle are important, since these may be tested on. Citrate synthase, for example, which is going to be inhibited by high levels of ATP, is going to convert acetyl-CoA with oxaloacetate to form a citrate molecule here. Isocitrate dehydrogenase is going to convert isocitrate to alpha-ketoglutarate, and this is producing an NADH in the process. Notice how we lost a carbon in the form of carbon dioxide as well. Now, it's logical since that it is inhibited by ATP and NADH. So it's going to be pretty straightforward that high levels of NADH inhibit this enzyme, and then also ATP. Remember, this whole cycle makes ATP. So if you have a lot of these, you don't need to keep going through the TCA cycle. Our last irreversible step is going to be the alpha-ketoglutarate dehydrogenase step. So we have 3 irreversible steps. Now, the alpha-ketoglutarate dehydrogenase converts an alpha-ketoglutarate into a succinyl-CoA. As I mentioned before, this alpha-ketoglutarate dehydrogenase complex uses the same cofactors as the pyruvate dehydrogenase complex up here. Do you remember what those cofactors were? The pyruvate dehydrogenase complex and the alpha-ketoglutarate dehydrogenase complex use 1. Vitamin B1. 2. They use vitamin B2. 3. They use vitamin B3. Hey, pretty easy. 4. No, they don't use vitamin B4. They use vitamin B5. And then lastly, they use lipoic acid. Alright, so that was a little side note. Do you remember what important synthesis pathway starts with succinyl-CoA? We just used our alpha-ketoglutarate dehydrogenase to create a succinyl-CoA. What can we use succinyl-CoA for as a substrate in the synthesis pathway? Now that would be the heme synthesis pathway. It's an important concept to remember. The TCA cycle is responsible for creating ATP, but it also is going to create intermediates for other pathways within the body. So succinyl-CoA is used in heme synthesis. Let's take a look at a flash quiz. At which steps in the Krebs cycle are NADH, GTP, and FADH2 produced? Give you a second to think about this one. This uses our TCA cycle knowledge here. This question is asking, where are the activated carriers created? Ultimately, the NADH is made at 3 main points. When the isocitrate is converted to alpha-ketoglutarate, when the alpha-ketoglutarate is converted to a succinyl-CoA, and then lastly, when the malate is converted to an oxaloacetate. The GTP is going to be produced, which is also an ATP equivalent for our sake. It's going to be produced when the succinyl-CoA is converted to our succinate. And lastly, the FADH2 is going to be produced when the succinate is converted to the fumarate. Now let's divert our attention to the inner mitochondrial membrane, where we'll find the electron transport chain. Now there are quite a few fancy figures out there, but I'm here to make this as simple as I can because knowing all of those minute details will not be important for the boards. However, the stuff that I'm about to discuss will be important. Now, In the electron transport chain, there are, count them, 1, 2, 3, 4, and 5 major complexes named complex 1, 2, 3, 4, and 5. Now, each of these has a unique function. Here, we see our inner mitochondrial membrane. If you can remember back to the mitochondria, there's going to be an inner membrane and an outer membrane. Let's consider up here the space between the membranes, so the intermembrane space. And then down here is going to be our innermost portion of the mitochondria. This is going to be our mitochondrial matrix. All right, so that's the anatomy. Our pre-made activated carriers. Do you remember the ones that I said that I kept saying they're going to feed into the electron transport chain? This is where we revive them. It's going to be our NADH. That'll be our FADH2. Those are our 2 activated carriers that contain the electrons. Now they're going to dump these electrons at these complexes to ultimately create ATP. So our NADH is going to enter at complex 1, and our FADH2 is going to enter at complex 2. Now, complex 2 is also called succinate dehydrogenase. This is an enzyme that's going to be found in the TCA cycle. So complex 2 is really just the enzyme succinate dehydrogenase, and that's going to be in the TCA cycle. So that enzyme is going to double as both part of the TCA cycle, which remember is found in the mitochondria, and the electron transport chain, which is found in the mitochondria. All right. So as electrons pass through these complexes, they create a proton gradient. And that proton gradient is going to be used to drive the production of ATP. So complex 1 is going to create a proton gradient. Complex 3 and complex 4 are going to create the proton gradient. Now notice how our protons are going to be pumped into this intermembrane space. Now, complex 4 is also known as cytochrome C oxidase, and this is going to transfer electrons to its final electron acceptor. Do you remember what that final electron acceptor for the electron transport chain is? It's going to be oxygen, where you get water as a final result, and complex 4 is going to dump its final electrons to this oxygen to create our free water. Now, without oxygen, there is no final electron acceptor, so this whole electron transport chain cannot function. And then the body must rely on anaerobic metabolism as a result. So you need this oxygen, that's why we breathe. Complex 5 is also going to be known as ATP synthase. It's going to use the hydrogen gradient that complexes 1, 3, and 4 make to produce 3 ATP for every NADH and 2 ATP for every FADH2. Now, remember I already said that NADHs make 3 ATP, while FADHs only make 2. Well, let's take a look. Remember I said complexes 1, 3, and 4 all make a proton gradient. And that proton gradient, so we have our protons up here, are going to be used by complex 5 to drive it down and make our ATP. Well, since FADH2 feeds into complex 2, it misses out on one of those steps. So it can't pump as many protons, therefore it can't make as much ATP. So it makes sense. NADH molecules can pump out protons in complex 1, so it can pump out more proton gradient, therefore making more ATP molecules. Now, our complex 5 is going to take a proton, and it's going to take an ADP, free ADP molecule, and a free phosphate to form the high energy ATP molecule. So it's going to combine these two, and it's going to drive that proton gradient, using that as its energy force to make ATP molecules. Alright. The electron transport chain inhibitors such as rotenone, cyanide, antimycin A, and carbon monoxide are going to directly inhibit electron movement from complex to complex. Now this prevents the formation of a proton gradient and therefore the synthesis of ATP. So rotenone is going to inhibit complex 1, antimycin A inhibits complex 3, and cyanide is going to inhibit complex 4 here. Oligomycin, on the other hand, is an ATP synthase inhibitor, meaning it's going to inhibit complex 5. So this allows the proton gradient to build up without any ATP synthesis. We're still able to make our proton gradient up here, but we're just not able to convert that proton gradient into ATP. And this inhibits electron transport and results in increased NADH levels and FADH2 levels. How do uncoupling agents work? Uncoupling agents such as 2,4-dinitrophenol, aspirin, and the protein thermogenin, which is produced in brown fat, increase the permeability of the inner membrane. So what we're going to do is we're going to create an increased permeability, meaning the protons that we create in our gradient are going to be able to pass through this inner membrane, bypassing this complex, and our protons can therefore flow through. Now this results in an increased oxygen consumption, since an increase in electron transport attempts to compensate for the permeability. So we're going to increase oxygen consumption because our complexes here will try and increase and compensate for the permeability. Still, ATP cannot be created without a proton gradient, and then thus the increased electron transport activity is instead dissipated as heat. This is why aspirin overdose can cause fevers, and why babies and hibernating bears can stay warm, since they have lots of brown fat. Okay, a 4-year-old girl swallows some cyanide-containing rat poison from underneath the sink. How does this affect her ATP production? It's pretty much saying, how does cyanide prevent her from producing ATP? Alright, remember that cyanide directly inhibits the electron transport chain, which decreases the proton gradient and therefore blocks ATP synthesis. Therefore, the body isn't able to create ATP via oxidative phosphorylation. So, in a nutshell, it's going to decrease ATP production by inhibiting the electron transport chain.